Hello, we are going to study about the reverse transcription PCR. In the reverse transcription PCR, here our sample is RNA. We use RNA and then we convert it into DNA, and that DNA we multiply into the multiple copies. The difference between reverse transcription PCR and normal PCR is that in normal PCR we use the sample of DNA from any of the places, let's say from the crimes or the research places where we're using PCR for the multiplying or amplifying any of the extracted DNA there. But over here, our sample itself is a RNA, and that RNA is converted into DNA, and then that DNA is multiplied using a PCR reaction. Since this process is involved with the transcription, that means reverse transcription, so we call it as a reverse transcription PCR, also widely known as RT-PCR. So in the RT-PCR, what is the basic things we need to understand in the RT-PCR is, now here converting the RNA to DNA requires a special enzyme, and that enzyme we call it as reverse transcriptase. And the amplification of DNA, how exactly once the RNA is converted, used as a complementary for the production of DNA, and we produce the strand of a DNA, that DNA has to be now we are having only single strand. That DNA has to again get the complementary DNA nucleotides, and that is used by the Taq polymerase. So basically Taq polymerase adds the nucleotides complementary to the existing DNA after the reverse transcription, and that the double stranded DNA then goes for the PCR reaction, and this is how exactly a normal principle of the RT-PCR works. Now where do we get where is exactly the RNA is present? As a time of a 2020, which was a year of COVID, which is also a RNA virus, so we know the RNA viruses have a RNA as a genetic material. So in this case, for the recognizing the sequence of the DNA or understanding the presence of the RNA, which if that RNA is present, we will give the positive detection after the PCR, after certain S's. So here's what happens. Now let's say a person is having HIV viruses, so that could be in the HIV the ELISA is also used. So these viruses RNA is extracted, and that extracted RNA is used for the RT-PCR reaction. So we have COVID, we have HIV viruses, we have Zika viruses, and the influenza viruses, which do have the RNA as their genetic material. So this is basically used, the RT-PCR basically used for the viruses which has the RNA as the genetic material. Now let's see how exactly a simple process of the reverse transcription happens in vivo, inside. So now we have a single stranded RNA template. We have a RNA template which is single stranded, so a long chain of a single stranded RNA template. So we have B and TPS here. Why? Because the nucleotides are required for the addition. So what happens now, reverse transcriptase, which is dependent on the RNA dependent polymerase enzyme it is. So it starts adding the dinucleotide triphosphates to the deoxynucleotide triphosphates to the RNA, a single stranded RNA complementary. So it's keep on add the complementary to the single stranded RNA, and now we have a one side of the single stranded RNA template, the other one as the DNA template, and this combination we call it as a RNA DNA hybrid. So now we have 5' to 3' direction here as a RNA, and we have the DNA in the opposite direction here. So basically this is how exactly reverse transcriptase has worked. The single stranded RNA has been now converted into a double stranded sample which contains RNA and the DNA combination, and we call it as a RNA DNA hybrid. In this hybrid, the RNA, the nucleotides, the RNA strands nucleotides are complementary to the deoxyribonucleotides. So once this double stranded RNA and DNA hybrids are formed, now we have a special enzyme. We call it as a RNase H. Why? Because we are converting our RNA into DNA. We do not want RNA. We are used RNA for the sample. So what does this RNase H does is it just breaks down it actually breaks down all the RNA strands. So the RNA strand is broken down into the individual nucleotides. Now we have again just a single stranded DNA. Here what exactly happens is the same thing what happens in the PCR reaction. So we are using this single stranded DNA has a complementary strand for the making a double stranded DNA. Here we use DNTPase, again deoxynucleotide triphosphates, but here the Taq polymerase enzyme works for the addition of the complementary deoxyribonucleotide for the single stranded DNA. So at the end of the reaction we have a double stranded DNA, which will be further we use it for the PCR reaction. So basically in the beginning we had the RNA template, which is a riboxynucleotides, and that RNA template we used, and we used reverse transcriptase enzyme. Reverse transcriptase enzyme is a RNA dependent DNA polymerase. That means it uses the RNA and then it adds the deoxyribonucleotides. So it added the complementary ribonucleotides to the RNA strands. Once the hybrid formation happens, the RNA DNA hybrid, so RNase H, the next enzyme, which will cleave the ribonucleotides, and then at the end of this we will have a single stranded DNA. And after this what happens is the DNTPase, again the Taq polymerase DNA, a Taq polymerase enzyme will work on this single stranded DNA and takes this DNTPase and adds the does the polymerization from 5' to 3' directions. So this is the entire reverse transcription process. In the reverse transcription process, sometimes what happens, we have a RNA. So if you see this right side picture, we have RNA, but in the eukaryotes the RNA has a poly A tail. So what is this poly A tail does is sometimes, so it acts as the is easy to use as a primers in the sense during the DNA replication we need primers for the starting of the replication process. So same here for the AA, the poly A tail, we will add the TTs, and which is acts as a primers. So that is what exactly in the eukaryote process happens. So what is the requirements we have in the basically we have RNA template. For the RNA template we use reverse transcriptase enzyme. We have primer, a complementary primer to the RNA is added there. So primers I will be speaking about it. Then we have complementary DNA synthesis by reverse transcriptase enzyme, which converts the RNA to the cDNA template. And then we have the RNase H, which will degrade the RNA template, and we will have only the single stranded DNA. So what are the ingredients for the reverse transcription? So we know our basic concept, we need a RNA template. So what are the RNA templates? You might be taking it from any person, or to find whether that person has the viral infection. We use the person's probably the pus or the mucus or any of the body fluids to understand whether that person, the area of infection, so where exactly it is present. Requiringly we will extract we will take that sample and extract the RNA template. So once we take the RNA, we have the reverse transcriptase. We require a reverse transcriptase for converting the RNA to DNA. So that is synthesizing the cDNA from RNA. So yes, complementary DNA for the RNA. Then we have primers. The primers, what are this is, even reverse transcriptase independently directly cannot add deoxyribonucleotides, that is DNTPase directly. So it needs something as a support. How exactly the Taq polymerase or DNA polymerase also does. So what exactly it does is, we usually use the short oligonucleotide sequences, or we have the random hexameric sequences, like the hexamers in the sense we will have a DNTPase which is in the 6 nucleotides in the different combinations. We tell them as a random hexamers. So these random hexamers will be used as the primers. So either if we know the sequence well in advance, we can use the short oligonucleotides as the sequences, but if that is not the case, we use the random hexamers which will act as a primers. So for the cDNA synthesis. Now we know after that we need a Taq polymerase. So Taq polymerase is here for from the DNA replication single stranded, we have to make it double stranded. So this is what exactly the ingredients for the reverse transcription PCR. So when I speak about the reverse transcriptase enzyme, so where does this enzyme come from? What are the requirements? Where exactly the reverse transcriptase enzyme come from, and what kind of reverse transcriptase enzyme I have to use? So that is a basic concept. So the one is we use it from the Molyneux leukemia virus. So basically this is a very commonly used reverse transcriptase enzyme. We extract the reverse transcriptase from this virus basically because it can make a high copy number, and that means it has a very good capacity to make a larger amount of the cDNAs with the existing single stranded RNA. So that is one of the reason why this is used. Secondly, we have a avian myoblastosis virus. So basically why this is used is because here we have a long strand of RNA sequence, like very long strand of RNA sequence, and we do not want a lot of copy. We want to have a very less copy, but we need to have all the entire sequence to be transcripted. So reverse transcript into the cDNA synthesis. So this is where exactly we use the avian myoblastosis virus. Then other types also we will use, but we should be specifically have a understanding what exactly kind of the reverse transcriptase enzyme we are using, and what are the purpose and the specificity of that reverse transcriptase enzyme. Otherwise sometimes it may not be able to give you the right cDNA synthesis, or your entire cDNA may not be reverse transcription, may not be synthesized. So that is what exactly the choices of the reverse transcriptase enzyme. Now we will directly come to the process of the RNA transcription. So this is the basically steps involved in the RNA transcription. The first one is the linearization of RNA template, annealing of the oligonucleotides. The third steps is the reverse transcription by reverse transcriptase, removal of mismatch by RNase H, mismatch repair by clinofragment DNA polymerase 1, and downstream processing of cDNA. So this is the total steps involved in the RNA transcription. So how exactly? We will go step by step for each step. So first we will go for the linearization of RNA template. Let's say you got a RNA sequence, or you have a RNA sample, a single stranded. But we know the single stranded RNA have some nucleotides which probably are complementary to each other. So what they does, they start linearizing. The linear structures may not be, they will be always in the secondary structures or tertiary forms. They will be complementarily bound to each other by the hydrogen bonding. So you may not get the single stranded sequence of the RNA. You will straight linearized RNA, rather you will be always getting the secondary structure of the RNA. So what we do, we simply give the temperature. In the RT-PCR, the temperature will be initially given to 50 degrees Celsius. In this temperature, the secondary structure of your RNA will be linearized into a single straight structures. So it may not be having any hydrogen bonding in between of that. So bases will not be bound to each other. So you will be linearized the structure will be having. So first step involves the secondary structures of your RNAs, which will be probably bound to each other by the complementary binding, will be linearized by using the temperature, which is 50 degrees Celsius. The next concept is annealing of oligonucleotides. Over here what happens, we need a primer, right? So we need a primer. So basically what we do is, whether we use the primers of the if it is adenine, we will use the deoxythiamine as the DNTP works as a primer here. So whatever happens is, like, we will have the complementary DNTP which is used as a primer. So usually if you know the sequence well, I mean if you know what are the sequence of the RNA, so you can use some strands or short sequences as the primer. So once you add the primer, if you are not aware, so there is another type of a primer we use, that is random hexamers. So what is random hexamers is, we know that is ATGC combination of nucleotides. We will mismatch the combination into 6. So we will have a 6 combination of these nucleotide bases, and these random will be, either one of them will be binding to the sequences will be combining to the sequences. So this is how exactly the random hexamers work. So now why exactly we need primers? Because we know our RT reverse transcriptase enzyme will not be able to directly add the deoxyribonucleotides complementary to the RNA. It needs the primer to start the reverse transcription, to synthesize the cDNA. So that is why we have a PCR. The PCR we use is either the one is a complementary to the adenine, we have it. If it has a poly-A-type. Other than that, we also have a random hexamer combination, random hexamer primers. Combination of 6 nucleotides, and other than that we also have the sequence specific primer. This is when you know the sequence. So if you know the sequence of the RNA, so I know this is the RNA sequence, you are very aware of that sequence, you use that specific sequence as the primer. So then both the cases of the primer, it is very random, that I am just predicting one of them might work. But in the third case, when I know the sequence of the RNA well, so any one of the combination could be used. Either you can use the oligo T as a primer, and either you can use the random hexamers as the primer, or you can use the sequence specific primer if you know the sequence of the RNA well. Okay, with that, once you know the sequence, when you are added your first sequence binding has happened by the primers, now your RT PCR will start, your reverse transcriptase enzyme will start adding the synthesizing the cDNA, complementary DNA to the RNA. So it has moved from the 5' direction to 3' direction. So the anti-parallel to the RNA template. So it is always moves from the 5' direction to 3' direction, and it starts adding the RNA DNA prime cDNA, complementary DNA to the RNA strands. So once this is done, we know at the end what happens. We have our hybrid RNA and DNA. One is DNA, one is RNA. Here the green is DNA, orange is RNA. So now I know I do not want this RNA sequence. I used it because I wanted to have a DNA copy from it. Now I have to make a double stranded DNA, but this hybrid, so I have to degrade this RNA. So here we will have a RNase H enzyme coming and degrading this RNA, and now I have a single stranded DNA. Once you have a single stranded DNA, we know that single stranded DNA, what happens, we have to make it double stranded DNA. So that is what exactly we are going to do. From the single stranded DNA, we are going to have a double stranded DNA. So how we are going to do it? So once the RNase H will be cuts the sequences, we will have the DNA polymerase also working there sometimes. How exactly is Taq polymerase works here. The Taq polymerase enzyme will add the nucleotide from the 5' to 3' direction using the complementary DNA. So at the end, we have finished the double stranded DNA. So at the end, we have got the DNA, double stranded DNA. Here Taq polymerase have added the double stranded DNA we have made. So from this now, once we got a copy of the double stranded DNA, we are going for the PCR amplification. What we studied previously, the same PCR amplification is happening. What is here is happening is your double stranded DNA is converted into a single stranded DNA by denaturation. So they are getting denatured. After that, the PCR reaction goes and the primers are used. One is forward primer, the reverse primer, and then one direction forward primer will be adding the Taq polymerase will be adding the complementary for the strand. The other direction again the Taq polymerase will be adding the complementary DNA. So this double stranded DNA acts as a template, and now we will make from this 2 strand we will make 4 copies. From this 4 copies we will make a 16 copies. So every time it is gonna be doubled. It's a geometric kind of a progression. So this is what happens. So here what happens, at the end we have amplified this DNA, and this sequences we are using for the any of the assays. So this concentrated sequences we use for the assay, like we can use it for the ELISA, enzyme linked immunosorbent assay. We can use it for the many other assays, like to find out whether that person has the has the infection or person has got the infection. So this is how exactly RT-PCR works. So it is a very good instrument in the medical diagnosis, at the same time in the research field, to understand the viruses and understand the structure, what are the RNA strands contributing for the viral functions, a lot of factors. So this is how exactly we will carry out the RT-PCR. So what are the limitations of the RT-PCR? This is very important, the quality of the RNA. So you should have a RNA specifically of one strand. Let's say you take a RNA sample from the person, and the person might be having the contaminated, your RNA is contaminated with some other RNA sequences. So that time what happens, you do not have a qualitative RNA, so you may not get the right result at that point of time. Enzyme efficiency, that is also a limitation. So what happens is, you're using reverse transcriptase. It is very important to understand which kind of reverse transcriptase you are using, whether you want a high copy of the cDNA synthesized, or you have a long linear if you have a long linear RNA, you have to synthesize end to end that RNA sequence. So understanding about what kind of a reverse transcriptase is used is also very important. qPCR validation, this is in the research, it is very important. So what is your PCR validation? So that means you should have a R square which comes around 0.999. So this is not very important, but for your understanding, for the theoretical knowledge, but PCR is very important. Validation is important in terms of a research analysis. The limit of detection. So sometimes sensitivity, if your RT-PCR doesn't have a proper sensitivity, or it doesn't have a proper temperature maintenance, or if you're not using the right amount of the enzymes or the buffers or the nucleotides, any of this, so it is always when you run the RT-PCR for the first time, you will never be getting your results. This is always the case in the scenario. When you're doing the first time RT-PCR or even PCR, you will encounter the problems in the steps, that this problem has happened, so let's change the one of the factor. So it's always like that. So limit of the detection is also works like that. So thank you. So with this I hope you've understood RT-PCR.